This is the detailed diagram of ammonia metabolism. I cannot find more simple diagram than this diagram. This figure shows both alanine cycle and a urea cycle. Now, before we discuss either pathways, perhaps it would be more helpful if I provide a brief overview first. Now, when proteins are catabolized or when proteins are metabolized, their amino group must be removed. And you guys very much know that, or are very much familiar that their amino group is released in the form of ammonia. And ammonia, as it is a toxic substance, it must be removed out of body and it must go to liver. In liver, it must go through a urea cycle and it must be released as form of urea and excreted in urine. So now keeping this in mind, now let's move toward pathways. I think it would be more helpful to discuss alanine cycle first on right hand side. Now alanine cycle again is a coordinated series of reactions between a skeletal muscle and a liver that help us or that let us use amino acid for the source of energy. Now this is not an ideal pathway, but this is the most useful source of energy during starvation. During starvation, we can use our muscles to create energy, to create glucose, and to create ATPs. So this cycle is very important for that purpose. So now let's look at the cycle. Now amino acid is converted to alanine. Amino acid transfers amino group to alanine. And this alanine goes to liver. And in liver, this alanine is converted to pyruvate, then glucose. Actually, in liver, alanine is going through gluconeogenesis, and in gluconeogenesis, glucose is formed, and this glucose is again used by extrahepatic tissues for the source of energy, as a source of energy during starvation. Right? Now you can see on the right hand side, this glutamate transfers its amino group to alanine. When this glutamate transfers its amino group to alanine, alpha ketoglutarate is formed. Now on this left hand side, you can see in liver, this alpha ketoglutarate is accepting ammonia, as accepting amino group, and is converted into glutamate again. Now we can say that this reaction is actually a reversible reaction. So this is actually a reversible reaction which is happening over here, and it is called as a transamination. Actually, this reaction requires some sort of enzymes called as transaminases. And for the activity of transaminases, we require a vitamin B6 or we require pyridoxine. So, right? Now, when alanine gives amino group to alpha ketoglutarate and alpha ketoglutarate is converted into glutamate, now this glutamate can transfer ammonia to urea cycle. With the help of enzyme glutamate dehydrogenase, this glutamate transfer ammonia, releases free ammonia. Now let's talk about urea cycle. That was alanine cycle. Now let's go toward urea cycle. Now what happens in urea cycle? This free ammonia, carbon dioxide and ATP, they combine with the help of carbamylase phosphate synthase 1. Same enzyme just like carbamylase phosphate synthesis 2, which we saw in pyrimidine synthesis. Now with the help of this enzyme, they are converted to carbamylase phosphate. Now this carbamylase phosphate combines with ornithine. With the help of ornithine trans-carbamylase. Now this enzyme is very important enzyme, and this enzyme's deficiency is a very common cause. This enzyme's deficiency is very common cause of hyperammonemia, a very common disorder which we see in liver. It's very common liver disease. So the deficiency of ornithine trans-carbamylase results in hyperammonemia. Right? Now hyperammonemia, as we know that the urea cycle happens in liver. So any liver disease or a urea cycle can cause hyperammonemia because ammonia will not convert into urea, so it will accumulate, causing hyperammonemia. So the common manifestation of hyperammonemia is a neurological symptoms or a neurological deficits. I'll tell you in the next slide that why neurological symptoms happen in hyperammonemia. Right? Now the common treatment for hyperammonemia include lactulose. Lactulose actually acidifies gut and it converts ammonia into ammoniamine, which can be excreted into stool. Right? This is a common actually treatment given in hyperammonemia, or sometimes we also use antibiotic. Right? Now why the hyperammonemia results in neurological deficit? आखिर hyperammonemia की वजह से neurological problems क्यों होती हैं, उसकी हम बात करते हैं. Now this is a very simple diagram जो मैंने इंटरनेट से उठाई है, very good diagram of glutamate glutamine cycle. So दिस इज़ द प्रीसिनेप्टिक न्यूरॉन. इन प्रीसिनेप्टिक न्यूरॉन क्या होता है, हमारे पास जो ग्लूटामीन होता है वो कन्वर्ट होता है ग्लूटामेट में. एंड यू गाइज वेरी मच नो दैट ग्लूटामेट इज अ एक्साइटरी न्यूरोट्रांसमीटर, right? It is very important, the synaptic transmission. It comes in synaptic cleft and help in a neurological transmission. After transmission, it is absorbed by astrocytes. In astrocyte क्या होता है, हमारे पास ये ग्लूटामेट दोबारा कंबाइन होता है अमोनिया के साथ और ग्लूटामीन बनता है. Now if अमोनिया accumulate in blood, अमोनिया की अगर जो कंसनट्रेशन बढ़ गई है ब्लड के अंदर, तो क्या होगा, अमोनिया ज़्यादा आएगा यहाँ पे, तो ग्लूटामीन और अमोनिया ज़्यादा कंबाइन होंगे और ग्लूटामीन ज़्यादा बनेगा. ग्लूटामेट और अमोनिया ज़्यादा कंबाइन होंगे, ग्लूटामीन ज़्यादा बनेगा. जब ग्लूटामीन ज़्यादा बनेगा तो इट विल रिजल्ट इन ऑस्मोटिक ग्रेडियंट. नाउ इट विल अब्जॉर्ब वाटर. एस्ट्रोसाइट विल स्टार्ट एब्जॉर्बिंग वाटर. When it will absorb water, it will be difficult for glutamine to be transported into presynaptic neuron. अब जब presynaptic neuron के अंदर glutamine नहीं आएगा, glutamate नहीं बनेगा, तो यहाँ पे न्यूरो ट्रांसमिशन होगी ही नहीं. So it will be defect in neurological transmission. So that is the key reason that why actually hyperammonemia results in neurological deficits. Now let's move towards cycle again. हम बहुत आगे चले आए. So we were at citrulline. We were actually at citrulline. We talked about that carbamyl phosphatase और एंथीन के साथ कंबाइन होकर citrulline बना रहे हैं. Now आपको यहाँ पे एक बात देखनी चाहिए कि this pathway till citrulline it was happening in mitochondria. Now after citrulline it will happen in cytosol. आपको ये बात याद होनी चाहिए या ये बात याद रखनी चाहिए कि यूरिया साइकिल दोनों पाथवेज को इस्तेमाल करती है, दोनों चीज़ों को इस्तेमाल करती है. माइटोकॉन्ड्रिया को भी इस्तेमाल करती है और साइटोसोल को भी इस्तेमाल करती है. Now in cytosol, citrulline will combine with aspartate with the help of arginine synthase enzyme and it will form arginosuccinate. Now this arginosuccinate with the help of arginosuccinate lyase will be converted to arginine and fumarate. अब इसने fumarate यहाँ दिखाया नहीं है क्योंकि it's very common, very simple diagram and I'm trying to keep things very simple. Right? Now fumarate will go into TC cycle, it will do its work there. But now arginine, with the help of arginase enzyme and water, it will be converted into ornithine and urea. Urea will be excreted by kidneys in the form of urine, and ornithine will again be recycled and will be used again in the urea cycle. So that was all for today. Thank you very much.